Hi guys, so nice to see you on the eve of your essay final. Are you excited?
No.
So there's no way I can possibly review, a whole semester in this amount of time, but I can give you some tips. I'm still working on your multiple choice, but your essay, you are going to have 10 questions, each of your versions. Let me look at the other one. You have 10 questions. Some of those questions are very short. Some of them are going to take a little more time. I would be familiar with all your labs so that you could talk to me how you would quantitatively set it up and justify your setup. Why would that be a good way to collect data, that type of a thing? And then whatever content was associated with labs. Because if you think about it this way, the labs that we do, okay, those labs are reflective, giving you an opportunity to take the content, right, and do more with it. And so, for instance, if there's a lab on cell membranes, you would want to study the membrane structure and function, to go along with it. Does that make sense? If we talk about photosynthesis and you have a lab on photosynthesis, that must be a big ticket item. Does that make sense to you? So connect the labs with, the topics that we're doing. Now, we didn't do our genetic engineering lab yet, because I do that in the spring when we have all the content done, because it's a really long lab. So that's not a lab you would have to set up, but that's, it's still an important concept, but don't expect any questions over any lab that you haven't already done. Does that make sense? Okay. I would be very comfortable with evolution. I would be very comfortable with chi-square tests. I'd be very comfortable with DNA structure, function replication, like DNA replication, transcription, translation. I'd be really comfortable with those kinds of things. Control of any of those types of things. When you lose control of any of those types of things, those are all big ticket items, I'd say. I'd practice doing a chi-square test. I'd look at the book that when I gave you the AP equation sheet, how I set it, they give you that. It's standard. It's nothing I do. But I would look at the way they have it so that you're familiar with it in case I use something from that AP equation sheet. Because I would give it to you exactly how they have it. Yes?
Do we get the AP equation sheet?
Or you'll give a portion off of it so I don't have to, so I have one less thing to hand you. Does that make sense?
Yeah.
I'll lift it right from it. Whatever you need, I'll lift it. Which is actually really nice because then you only have to pay attention. You know what you have to pay attention to. Yeah. I'm going to just give you a heads up now, just because tomorrow you'll be busy essay writing the whole time, right? I'm working on your multiple choice right now. I have lifted, if you look at some practice, I know some of you are looking at practice exams, right? Real practice exams from the College Board, right? You buy booklets or whatever to help you with that. They will have like a whole scenario. You could have to read a whole page and then answer 5 to 6 questions on it. Be prepared for those kinds of questions because it's a mindset. You know what I mean? You're either going to approach it, and that part you're doing individually. You're either going to approach it and it's going to terrify you, right? Or you're going to approach it and you're like, okay, I got to focus. The downside is this. These are all going to be fresh photocopies because I'm writing it right now, but I may want to use it in the future. I don't know. So you can't highlight and bold, but when you take the real AP exam, you'll be able to like circle things and underline. So you just don't have that benefit with me. You would want to do problems even on the Scantron part. You can bet that I'll have you work some problems out and just write them on the back of your Scantron. You know how you've seen Scantrons before, and on the back they have an open spot. So everything won't be a bubble answer when you do your Scantron part. I'll have some open questions that you have to show me the math for on the multiple choice part. So I have 50 questions, so 50 times 2 is 100, so it'll be 2 points each on your multiple choice. And the question on the 10, I told you it was 10 questions, right? I asked you on the essay part. You know, some are worth like 3 points, some are worth, 10 points. It depends on the type of question. That's very typical, on your AP exam too. You'll have, except they set it up where the biggest points are at the beginning and then it tapers off. But we won't worry about that right now. What is that?
It's a headphone. Didn't turn out.
Oh, I didn't get to see the face. Okay, yeah, that helps. I'm just gonna keep to myself. Yeah. Okay, so those are my strong hints that I'm giving you right now. Is there anything you would like to ask me? Is there anything, not what are the questions, but is there anything you'd like me to review with you? Yes.
Should we be familiar with the highly suggested reading?
No, you should not. Yeah. Let me just verify that before I say that. I'm just going to verify. I don't think so. Let me just verify. I don't want to lead you astray.
The most recent chapters I kind of know, but like the ones from like no. Like chapters from 9 are about where we are. I'm familiar with.
Yes, I can say that with authority. Is that better? Okay. Good question. Yes.
I just have a review question kind
Sure.
of based on like the Unit 4 exam. There's like a lot of like applying the evolution like, terms and stuff to certain scenarios. Could you just kind of like go over those kind of scenarios? And, like,
Which ones? I'd be happy to talk about anything.
I'm trying to think of like I don't remember like specific questions from the test.
Just give me like an idea of something and I can run with it.
Oh, it was like what are examples of like the bottleneck effect, and if you had two like adjacent populations, like what?
Oh, sure. We can talk about all that. Sure. Okay, so just to make sure we have the big ticket item, and then just say stop, Miss Litton, if you totally understand this and you don't want me to talk about it. Or say yes if you want me to keep going. The big ticket item on Hardy-Weinberg is, keep in mind, Hardy-Weinberg wants you to understand the inevitability of evolution, because anything that alters allele frequencies causes microevolution. And the 5 big things we discuss can cause that change in allele frequency or microevolution. And if enough of that happens, that can lead to what? Macroevolution, which is speciation. Good. So when we look at those 5 things, the opposite of that you have to do in order to maintain allele frequency and no evolution. So one of those things when we watch the video of the 5 fingers, I think that's the most helpful for you to remember it by, is small populations. And small populations are more prone to genetic drift, because losing a few in a small population has a bigger impact than losing a few on a larger population, right? So of your things in genetic drift, there are 2 categories of genetic drift. And so one category of genetic drift says, this is my original population. You can tell me what category this one is, okay? This is my original population, and a group of them went over here and started another whole population. Okay? And because this population was so small, and a group went over here, and now you only see the greens and the blue, what would that be an example of? Founder's effect. Okay? So in a situation where you have a new colony established with a small fraction of the original group, and it's not representative of the whole. Okay, what's the other genetic drift concern we have? Yeah, so bottleneck is when they go through a what? Yeah, some sort of disaster. So if we have a disaster and all of a sudden a flood comes up and kills some of them off, okay, then that's an example of a bottleneck. Only a few survived, and those that survived were not representative of what that community was. So that was genetic drift. Then there was another one that could cause changes in a population was non-random mating. So in non-random mating, you're choosing particular traits and you're selecting for those. You're only still going to be able to, you have the same deck of cards. You're not changing what's in your deck of cards, right? You're just making more of this group be together and more of this group be together, which ultimately could lead to speciation, but it's not really changing your allele frequencies, right? Wiping out some genes like this in genetic drift could, right? That could change your allele frequencies drastically. What's the middle finger? Mutations. So that's mutations to a whole new allele frequencies. And what they say is, you could say, the opposite, what's the opposite of mutations? No mutations, or another word for that is called mutational equilibrium, which means as many mutations of A to B, you have just as many mutations from B to A. So that's mutational equilibrium, right? So that could lead to new alleles, right? And then what's the pointer finger? Migration. So migration would be going back to this situation right here, okay? So migration could be that I'm bringing in a whole flock of new alleles into this situation, right? And that would be gene flow, right? Bringing in some new trait. Or if some of them migrated out. And the last one was natural selection. Yeah, natural selection. So, you know, things could change, the environment could change, and some traits could be selected for or against. Does that help you? Or did I hit your...
There's just one like specific question on the unit 4 example. I can't remember like exactly, but it was something about like if two species are like next to each other, like what would occur?
Okay, so let's talk about other forms of speciation. So there's allopatric speciation, and there's sympatric speciation, and there's adaptive radiation, right? So allopatric speciation, you have some sort of geographical barrier usually that you go around, and you want to make sure you address when you have some sort of geographical barrier. Is this somebody's mother out here? You have some sort of geographical barrier. Is it anybody's mother in here? You need to go talk to her? Okay. I want to talk to you now. Some sort of geographical barrier that separates you, and so then you have different initial gene frequencies, different selection pressures, different mutations that you accrue changes, and over time if you come back you're still going to be two different species. Sympatric speciation, you're side by side. You have a change in chromosomal number usually that leads to sympatric speciation. It could be a change in microhabitat, but generally speaking it's going to be a changes in chromosome number, which works fine for plants and not for animals. And then the last one, adaptive radiation, is where you usually have one ancestral species that encroaches out into other habitats or other niches out in there, and you get selected for certain traits, and it spreads out, but it all anchors back to one ancestral species. Not like, you remember the different kinds of selection? There was directional selection. Remember, I think I gave you a pogo on that, which would favor like longer neck giraffes over shorter neck giraffes. If you either should be a brown beetle and be on the underside of the leaf and stay hidden, or a greenish beetle on top keeps you hidden, it's a, you know, you want to be brown or green. You don't want to be a mixture of brown and greenness. What would that be? Disruptive selection. And could that lead to speciation? And could directional selection lead to speciation? Okay, and what's the third one? Stabilizing. And does that lead to speciation? No, it favors the norm. Okay, what else? Yes?
There was a question on the study guide that I didn't really understand. A linear strip of DNA.
Yeah, got it. I remember you showed it to me. Yeah. So if you had a string of DNA, here it is. It's the short answer questions that I gave you. It's number 11, okay? And I have 5 enzymes.
No.
5 cuts, 5 recognition
Two
sequences?
restriction sites for the restriction enzyme ECO R1.
Okay,
and 3
wait, listen to it. You
And-
have two recognition sequences. 2 recognition sequences for ECO R1. Keep going.
3 restriction sites for the restriction enzyme kind 3.
Hindi 3.
Yeah.
Yeah.
If a strip of DNA was incubated with the restriction enzymes and the cut DNA was collected and run on an electrophoretic gel, how many bands would be expected on the gel?
Okay. So if I have, remember what recognition sequences are? That's where restriction enzymes will bind onto the DNA where they can recognize it to cut. So I have 2 for this one. So here's 1 cut. I don't know where they are, right? There's 1 cut, and then here's another cut, right? And then I have how many? 3, 4 Hindi 3. Here's 1, here's 2, here's 3. Are we good? So how many fragments do we have there? 6. Okay, because I had 2, 1, so this snipped this off. 2, that snipped that off. 3, 4, 5 cuts. So I have 1, 2, 3, 4, 5, 6 banding patterns, and I should see on my gel. I don't know how big or small they'll be, but I would now have 6 fragments. Does that make sense?
Yeah.
Okay, what else? Yes.
There was one on the same document that this one was on,
Mm-hmm.
but it's about half-lives. I'm trying to find it. Oh, it's number 13. It's like the radioisotope potassium-40 can be used to date past events older than 60,000 years.
Is it P? What is it?
Potassium-40.
I know, but what is it short? Is it K?
Yeah.
I think
It's a K, right?
Yeah.
K40.
I was like, wait, what?
Is it a P? Is it a K? I can't remember. Yeah, K40. Okay, go ahead. I'm sorry. I'm
It's
listening.
okay. Potassium-40 has a half-life of 1.3 billion years, decaying into argon-40. Argon-40. If the igneous rock layer that scientists wish to date shows a ratio of potassium-40 to argon-40 at 1/4 the current ratio, what is the age of the rock layer?
Okay, so it's at 1/4, right? So 1 half-life would take it down a half, right?
Yeah.
Right? Another half-life would take it down a 4. So 1 half-life is 1.3, another half-life, because this would take it down a half, right? Then another 1.3 billion years would take it down another half, which would be a 4th, right? A 4th of it would, because I keep taking off half.
Yeah.
So that would be a total of, that thing is 2.6 billion years old.
Okay.
Does that make sense?
Yeah.
All right, what else? Okay, yes.
Could you do like I feel like for DNA replication, like I have a lot and I don't have it really condensed
Sure.
into.
Want to just talk about it?
Yeah.
Okay. Okay. If I was going to do DNA replication, what would it start with? Okay, so I have to do unwind and unzip. What's that? Yeah. Helicase, topoisomerase. You'll get that, right? Unwinds and unzips. Now I have how many strands? 2 strands, okay? So this will result in 2 single strands of DNA. I've unwound, I've unzipped, right? Now I can go, if this is a linear chromosome, I can take off in 4 different places, right? If I'm starting right here, I can take off that way. On the other side, I can take off that way, right? We're okay with that? Okay, so if we looked at it, a lot of times in your images, and I know that's why it gets confusing, is you'll see it like this, and they'll have an open strand. So they're working and unzipping right there. They're unzipping it, right? And on your leading strand, as it's already taken off, this is the progression of which way it's going. Okay, so that means I started somewhere over here, right? And if this were the leading strand, I would make complementary, and I'd just be going da 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 da, making my complementary, right? But before I can take off, I need to have, before I can use my DNA polymerase 3, I would have to lay down some what? Primer. I would have to lay down some primer. So I'm continuing to unzip in this way, but I started right here. And when I said I could take off in 4 directions, keep in mind that I could also take off in this direction, right? And I could also take off, if I started right here, I can go off in all 4. So if it's this way as the leading strand over there, then where would the leading strand be over here? The top one or the bottom one? Bottom one. Right? Does that make sense? Okay, but what did you tell me? I had to put some what down first? Primer. Okay? I would have to put some primer down. And who puts down the primer? What's the name of the enzyme? Yeah, RNA primase. And that's how you're going to keep it straight. Is that, remember, anything that ends in ASE is an enzyme, right? So you're going to put down primer, but the thing that's going to be putting down the primer is primase. And the primer is RNA. And how long is the primer? 10 base pairs long. Okay, 10 base pairs long. So our first thing is helicase and topoisomerase. Then the next thing we're going to use is RNA, and you would tell me that, RNA primase. And it will lay down primer. 1 time on the leading strands, right? I'm not writing it all the way out. And multiple for the what? Lagging strands. Good job. Okay? Once I have, yes?
I just have a quick question. Wouldn't you have to, in between helicase and RNA primase, wouldn't you have to lay down SBB or the
SSB holds them apart. Yeah, let's put that in there. It's not an enzyme, but SSB will keep the strands apart. SSB, perfect. Okay, so I want to put it down multiple times, and the idea is if this was the leading strand, this was the lagging strand, right? And so I have to backstitch. Oh, let me pick the right color. I have to backstitch and put some primer, and then backstitch and put some primer, and backstitch and put some primer down. And then off that primer, then that DNA polymerase can work, and it's DNA polymerase what? 3. DNA poly 3 will take and bring in complementary nucleotides to the strand that you're working off of, right? A and T and G and C. And I would do that on the other side as well. Then what's the next enzyme I need? DNA poly 1, which will do what? Remove the primer and replace it with DNA, complementary DNA nucleotides. And then I need to stitch those together. So what am I going to stitch that together with? Ligase. And it has to hook together, you know, the phosphate sugar, the sides of the ladder, right? The phosphate sugar backbones, it's got to stitch that together. Now you probably want to throw in there for me is, we know what these things are called right here in between the primer. So what are those called right there? Okazaki fragments. And about how long are those Okazaki fragments? 1,000 to 2,000 base pairs long. Yeah. Okay, did I answer that question? Are you okay with that? All right. Yes. Sure. So for translation, you want to talk transcription and translation? Is that what you're asking? Okay. So, what is it that you're really asking me? Okay. So, let's. What you're asking me really about is called the central dogma. Central down dogma. Okay. So tell me the 3 parts of the central dogma. Oh, I don't like that. Okay, that's terrible. What is that supposed to be right there? DNA. Okay? So DNA to more DNA is called what? Replication. Okay, what's the next part of the central dogma? Yes. DNA to a strand of RNA is called what? Transcription. And then using that RNA to build what? Proteins is called what? Translation. All right, we good with that? Okay, so what's different than what we learned in replication is you still need to have the DNA unwind and unzip. But now, since RNA is single-stranded, I don't need both strands, I just need one. So which strand am I going to use, the leading or the lagging? I'm just going to use the leading strand, and instead of using DNA polymerase, what am I going to use instead? RNA polymerase. And yes?
Do you need helicase and polymerase to unwind DNA?
You can mention that, but nobody really talks about it.
Okay.
Yeah. Yeah, I mean you can, but yeah. I don't even look at that. If somebody writes that, I just read over it. So then you're going to bring in RNA polymerase, and you're going to boom, boom, boom. But this time, and this is what you have to be very careful about, you need to tell me what you're specifically doing. Do not assume I know what you're talking about. I do know what you're talking about. But one of your science practices is being able to communicate effectively. And just because I know you and I know how you write, remember, you're not going to be writing for me in a few months. You're going to be writing for a random person that doesn't love you and doesn't care about you. I'm sorry. They don't pray over you like I pray over you. They do not love you like I do. So they're not going to take the time to reread and try to figure out what you said and, and go work for it. They're just going to go, was not effective, no. So you need to practice that right now. You need to talk about RNA polymerase bringing complementary RNA nucleotides. RNA nucleotides are different from DNA nucleotides because it has the sugar ribose instead of deoxyribose. And you do not find the base thymine. Instead, you find the base uracil. Yeah. And so you make complementary RNA. And you're going to copy, if it's a eukaryotic cell, it's going to have both introns and exons. And it's going to make a copy of all of it. And to protect, it's reading 3 to 5, making complementary 5 to 3.
So you don't use polymerase?
No. And it's going to make 5 to 3, and you're going to put on the head, what are you going to put? 7-methylguanosine, which is your cap, which protects the end of it, right? And what are you going to put on the other end? Tail of what? Adenines. How many? 100 to 200 adenines. That's going to be your tail. One. I just put one. I have a tiny tail. So where am I doing this? If this is a eukaryotic cell. I'm the nucleus. Okay. So now I made my mRNA. There's more details to that you do not have to, hairpin loop. You don't have to know all of that. Okay. Just be able to make that you made your mRNA from your DNA. Yeah. Okay. Now it has introns and exons, and I need to only pay attention to the exons. I could have the same DNA that codes for multiple proteins based on how you process it, right? So you're going to remove some of the introns because the ones you express are called the exons, and you're going to use what? SNRPs, spliceosomes, yeah. Okay, and so you're going to remove your exons, and now you're ready to leave the what? Nucleus. Now your mature RNA, and you're going to go through the nuclear pore. That is transcription. Translation, right, is when you first hook up with a small ribosomal subunit and then the large. And you actually technically don't hook up with the large ribosomal subunit until the first tRNA1 lands. Where does it know how to land? It's still working on things being what to each other? Complementary. And we talk about codons and anticodons. Who has the code? mRNA, because M stands for, where do they get the message from? DNA, right? So every 3 bases on the mRNA is called a codon, and it's complementary to an anticodon on the tRNA. Okay? So once tRNA1 lands, and it's always going to be AUG, and it always codes for methionine right off the bat. Then the large ribosomal subunit binds on. Now along comes tRNA number 2. And he lands right next to tRNA number 1 because he's in the middle of that ribozyme, and that's why we talk about RNA being the first genetic code, because it can act as both storing of genetic information. It can also act like an enzyme, right? Okay, so then tRNA1 dumps off his amino acid onto tRNA2, and the ribosome makes the peptide bond between the amino acids. It facilitates, it acts like an enzyme to facilitate that peptide bond. Then RNA1, or tRNA1 exits. He's now, as the ribosome shifts, he gets shifted from the P spot into the E spot. And now the tRNA that was in the A site gets moved into the P site. And I think of P for protein and A for amino acid, but there's other names for it. You won't have to write those out. If it starts with an A and they say a site, then you go, oh, that's it, okay? Then tRNA, what are we on now? 3 lands, right? And tRNA2, due to the ribosome shifting, dumps his amino acid over onto tRNA3, and then he's in the E site, and then he leaves. And this is going to go on and go on and go on until you reach a stop codon. But once you've done that, the protein's not done. It has to be what? Folded. Parts of it might have to be removed. Who's going to do that? Chaperone. And that talks about control, right? Because we look at all the levels of control. It starts way back in the nucleus. Are you heterochromatin or are you euchromatin? And that's epigenetic inheritance. I could have the genes to code for brown eyes, but my eyes appear blue, absence of pigment, because the gene for brown eyes was tightly wound and it was heterochromatin, it wasn't expressed. Okay, I had the gene, but it wasn't expressed. That's epigenetic inheritance, right? And then number 2, we can talk about transcriptional. Yes?
Would epigenetic inheritance be more commonly found in females? Because there's 2 X's.
No, you wouldn't say that. You would just, no, you wouldn't say that. Yeah, 'cause that's only one of your 23 pairs. Yeah, so that's something that you can pass on by the way you've tagged your chromosomes, and then it gets tagged once you've made a during the DNA replication part. You can end up tagging both sides so that the same ones are going to be tightly wound or uncoiled depending on what it is. Yeah. Okay, then transcriptional control, as whether RNA polymerase can even get onto the DNA, whether it's positive, negative, whatever. Post-transcriptional control is about and capping and tailing and exiting the nucleus. Translational control is whether or not you can get onto that ribosome and build that protein. Post-translational control is all the folding. Okay, what else? Yes?
This is a really quick question. It says predict the effects that introduction of free oxygen in the atmosphere had on the prokaryotes. Is it just that they now became
Wait, did you all hear her question?
Yeah.
Predict the effects of free oxygen in the atmosphere and what effect they had on prokaryotes. And you're saying, is it just that?
They became like aerobic, or were able to.
There's more effects to it than just being, they could do aerobic respiration, okay? That's true. And aerobic respiration generates more ATP, more bang for your buck. So now they could afford to go from being unicellular to being what? Multicellular. But there's more to it than that. Also having oxygen out there created an ozone layer, and it allowed you to move out of the water onto land. And number 3, it made it so no other creatures started evolving abiotically, because it was an oxidizing environment that broke down instead of a building environment. Mm-hmm. What else? Oh my gosh. Yes.
Didn't the introduction of oxygen also kill a lot of the prokaryotic cells?
If they were, if the only way they survived was being anaerobic, yes, it would kill them. If they had to live in it, you have facultative anaerobes, you have obligate anaerobes. Have we gone into all that? Obligate anaerobes, they have to have no oxygen in the free environment.
Okay.
Yeah. Yes?
This is a little off topic, but can we send your tests tomorrow at lunch or after school?
Is there some test I haven't made available to you?
Um,
I thought I made all of them available to you at this point.
Unit 4, that was
Unit 4, you never got to look at? Sure, you can look at that. You can look at it if you finish your essay early. Yeah, sorry, I thought you got to look at all of them. You can, yeah, no problem. When's my help tomorrow?
I think you're asking for
I think it's honors bio like open question.
Honors, yeah. Okay. So, and am I doing it lunch or 7?
Probably lunch.
I'm pretty sure
Usually
Monday-
Fridays, but because of finals, I didn't leave early any day this week. I just stayed after school. So, 7th period, or do you want to do it at lunch? Doesn't matter to me. Sure, lunch. You got it. Or if you finish your test early and you want to look at it, you can do it then too. That's cute. Okay, what else? I'm sorry, I thought you got to look at everything. My bad. I'm sorry. Okay, what else? Definitely want you to look at it. I always want you to overlook my math. What else? Anything? Is it important to sleep tonight? You guys, you need all your cylinders working, and when you get exhausted, you crash and you hit walls. And then you're just like, tonight the night to break up with your boyfriend or your girlfriend? No. It is tonight the night you go down the rabbit hole of watching YouTube videos? No, unless they're scientific. Wait, and 2 more questions. One more.
Okay, what is the microRNAs and
Small interfering RNA, or microRNAs, because RNA is single-stranded, it can interfere with other single-stranded, which is always all RNAs. So you could have some nice mRNA ready to be translated, but a small interfering RNA might bind to it, and now there's no way for the tRNA to land. Right, just logic. You know, I can't land because now all of a sudden single-stranded RNA became double-stranded. Does that make sense? SNRs are the ones that's part of removing the introns. All right, make good choices. I see you mañana. Yes. Thank you.
So are we going to